All right, everyone, I'm going to start. Let's see. Oh, so for the, we're in the last part of the class, right? So I'm going to be teaching for the last 2.5 weeks of this. So if you look up on Moodle, their PowerPoints for the last 3 classes should be up there. Between Friday's class will be a mixture of probably the end of this speciation talk and then probably a beginning of the phylogenetics talk. Those 3 are gonna the 3 PowerPoints I have on will be spread out for the last 4 classes. Quick update, if you had looked at your syllabus, it said that your final for this class is a cumulative file. We're not gonna be doing that. It's just gonna be on this unit. So your final is just gonna be a unit quiz on everything I teach for the last 4 classes. The last class also is set to be a synthesis day. So I'll give you it says that it's not sharing. When I was talking before, the people on Zoom, were you able to see the screen?
Now it looks better.
Oh, okay, great. Thank you. Thank you very much. All right. All right, so that last day would be a synthesis day. So we'll do some type of review session and I'll also give you some type of reading quickly. We'll probably talk about global climate change. I think I found a pretty good paper, a pretty good thing. That's pretty cool. Right now, the government is doing this thing where they're actually breeding specialized mosquitoes and they're trying to release those into the environment in order to cull certain species of mosquitoes that carry, a higher rate of malaria and other types of drugs. And so we'll talk about environmental impacts of things like that and kind of discuss, how we affect our environment. Because the end of this unit is going to be the end of speciation. So we're going to try to tie together all these concepts that you've been talking about in terms of evolution and genetic change. So we'll tie those together and then talk about phylogenetics, or how we put these ideas together and try to plot essentially how organisms have changed over time. And so, so in this, we're going to be doing essentially a conceptual bridge between micro and macroevolution, right? So how do we notice these small changes in genomes? How do those result in new species, and how do those species slowly change into huge new groups, such as, the transition of from dinosaurs to birds and things of that nature. Does anyone have, before I start, does anyone have any questions about how the last unit of this is going to run? No? No chat questions? Okay, cool. So, from what we've been the final is going to be on Moodle, like the others. We're going to give you, generally, I like to give you like 24 hours in order to take it. So it'll all be on Moodle, and so we'll just do it there. Cool. All right, so all right, so does anyone have a nice broad definition for what species is? Does anyone think they're comfortable explaining what they think separates one species from another? No? All right. Well, that's okay, because that is a very complicated thing, and that's what part of this is going to be. If you did your reading for class, this first part of the lecture is on species concepts, or how different scientists like to look at an organism and decide whether or not it's a new species. So one thing you're going to learn, something I try to highlight, is that this is a very complicated thing. figuring out if something is a new species is not trivial, and in fact can be very, very difficult. So for instance, if you are trying to find a new species, let's say you find a spider or some type of organism, one of the things you have to do in order to figure out if it's a new species is you have to document its different parts of its lifestyle, you have to document its physical plan, but then you have to go to a set location. For us, it would probably be the Museum of Natural History, in which they have thousands upon thousands of samples and preserved bodies of organisms, and you have to check your organism against all of their samples to make sure that it actually is novel in some way. And so the reason that this is complicated is because we've talked about how there are genotypical and phenotypical differences between organisms, and having the same genotype can result in different phenotypes, right? And so this definition can be very, very complicated. So the definition that you're probably most comfortable with and the one that's used most broadly in science is this idea of a biological definition of species. And so this is also, I'd like to think of more of an ecological aspect. It's kind of what we can notice in the environment. And so a species is a group of individuals, so a population of animals, generally speaking, who only mate with one another. And so there can be many different reasons that they only mate with one another, but this concept is known as reproductive isolation. Either they are not physically near one another, or they just don't have the right equipment. But either way, once that population starts to isolate and only reproduce within itself, does it start to become a new species? And so one of the other important things we'll talk about is the idea of having viable offspring. And so there are a lot of animals that we can mix together. Maybe they won't mate in nature. We do things, I'll talk about hybrids in a little bit, such as, ligers and tigons. We can mix the DNA of closely related organisms, but they may not necessarily be able to propagate or continue having offspring. Often hybrids, I'll talk about, are going to be sterile. And so those don't lead to a new creation of a species. Those are more of an evolutionary dead end. And so this is the basic definition that we have. But what I'm going to go over now is just showing you ways in that this can be very complicated, because the way we reproduce is very different than, say, asexual organisms or bacteria, right? If you take a bacteria and you try to talk about reproductive isolation, something that reproduces asexually, you've already broken this definition. And so it's going to get a little difficult. So as a quick start, I just want to talk about the ideas of species versus breeds, because on this screen right now, I have two examples, right? We have betta fish, which are these beautiful fish that have been bred. If you actually look at them in nature, betta fish are naturally browns and greens. If that makes sense evolutionarily, if you're sitting in a pond filled with grasses and weeds, you're probably just going to be some color similar to that, right? But we have specifically bred and forced these betta fish to not only be those colors, but we've highlighted and forced, reds and other colors. The most classical example of breeds would be dogs, right? If you look at all of these types of dogs, if you think of every dog you've ever owned or met, all of those dogs are the same species. You may not physically be able to take, a Chihuahua and breed it with a bull mastiff or one of those other huge dogs. But if you were to take their eggs and their sperm and combine them, they make viable offspring. And so breeds are an example of human forced evolution, as opposed to the idea of natural selection. And so breeds are morphologically distinct members of the same sex. Of the same species. And so they produce non-sterile offspring, and they're genetically, if not physically, able to copulate. And so it could be confusing because, once again, breeding is coming from genetic isolation and selective breeding, right? But this is not something that's occurring in the environment. this is something that we're forcing. And so this is our own selective breeding. And so theoretically, this is kind of, this is how natural selection works in concept, but just as a note, breeds are just the same species, just physically different. And so when we're talking about complications of this definition, one of the things that's important is we're talking about reproductive isolation, right? And so one of the problems is that we cannot possibly observe the mating behavior of all creatures, right? There are certain species that we've identified in the world where we've only found a single one of those organisms. Sometimes we don't know what the male or the female of a particular species looks like. And so there's that issue, but then also there are organisms who are interacting with each other in an environment. We're not constantly watching that bird. Is that bird really only mating with other sparrows, or is it occasionally, confusing a blue jay for a sparrow or something along those lines? But what I'm going to talk about first is this idea of these asexual organisms, right? But like, how biologically isolated are these creatures? And so we may never know, depending on the species. We can't observe organisms that are living in the deep sea all the time, right? How many live giant squid have we found? Maybe one. Usually they're dead and washed up. So how do we tell what those species are? But again, even in the most basic way, asexual species are going to be extremely complicated in order to diagnose, right? And so have you seen these? Starfish. Starfish reproduce asexually. They can split in half. In fact, depending on the species of starfish, if you were to cut up the starfish into 5 sections and you left at least a little bit of the center stomach with these organisms and you threw them out, you would have this starfish start growing into 4 other organisms. And so how do we define a species under these contexts? And so, not only are starfish doing this, but we have microscopic organisms who are obviously doing this on a much greater scale. So for instance, bacteria can share DNA with one another. They can do gene transfer. And so you can, that's why we have a problem with antibiotic resistance. We have bacteria, which are just sharing resistant genes with one another. And so a bacteria could absorb a new DNA plasmid, something along those lines. And then it becomes some type of genetic hybrid, right? To a small extent. It's still going to be reproducing asexually, though, right? It's still going to be reproducing with itself. So how do we decide that species A, say, Staphylococcus, the bacteria that causes a staph infection, how do we decide that Staphylococcus is a new species, when it's just gotten an resistance to penicillin? And so what they tried to do is scientists try to look at what makes a set of organisms different than another in their environment, right? And so we consider, I don't like this. Cool. So it's considered a different species when it fills a different environmental niche. And so in terms of bacteria, that could be something such as, breaking down different materials in the environment. All of a sudden, those bacteria have developed a new plasmid, and now they're no longer, undergoing, respiration. They're now doing photosynthesis, or they've now changed where they're living. They've spread a little bit more, and now they're being eaten by a new organism. And so when they fill a different niche in their biological processes is when these scientists decide that these become a new species. The thing with that is that, there are other ways to complicate this. So for instance, fossils. Fossils are how we know a lot about everything starting science, right? Starting written history, burn the Library of Alexander. We have no idea how much information they had about the past and their fossils then. But as of now, we're analyzing these and we're trying to look at how organisms are changing over time. And so because fossils are found in random places, random times, not every organism is going to be encased in some type of, amber or mud or something that will preserve it well enough, right? We have decomposers, we have things that are breaking down in the environment. And so this is a huge limitation on, again, those ideas of reproductive isolation, right? If we're looking at a fossil and we're only seeing bones, is it clear when this transition between species happens, when an organism is no longer able to mate with what it came from? And so this becomes clear and almost impossible depending on the level. Obviously, if something lived 10 million years ago, it's probably a new species. But when you're looking at fossils compared to 9 and 10 million years ago, does that organism just have an extra claw because it has a mutation, or is that actually leading to a new species? And so the other thing is that when we're looking at fossils, very rarely do we find single-celled organisms within those fossils. There was actually a new study that just came out in which, or not even study, but an article where they were talking about how they actually just found what they believe to be the first multicellular organism, and it was actually frozen in amber and fossilized. And it was impressive because usually these organisms break down. They're very simple, right? You have just that cell wall. You have, a bacteria that's going to be eaten or degraded by something else. And so things that aren't fossilized and single-celled organisms are missed. So we have to, again, make assumptions about the history of these organisms. And so the other thing is that, again, this is not as simple as looking at physical differences with organisms. So for instance, there's something known as phenotypic plasticity. And so certain organisms, depending on their environment, will actually show different phenotypes with the exact same genotypes. So I believe that Professor Strauss went over this. There are environmental factors which will actually trigger differences in genetic makeup. So for instance, certain flowers will change the coloration of their petals depending on the acidity or the basicness of the soil that they're in. And so these different environmental cues can affect what they look like. And so, for instance, these two insects here look very similar in body plan. But if you notice, this one on top actually has wings on it, right? And these are actually, they're not a different a different sex from one another. They're the exact same species. They're the exact same. They're both females, I believe, the exact same sex. They hold the same niche within their species. But depending on the environment, aka in this case, the leaves that these organisms are living on, whether or not they are healthy, whether or not they have a good environment in terms of, is this tree alive? Is this tree dying? They will actually change their physical features as they're growing due to these traits. And so the changing of morphological and behavioral traits to an environment is something that's common, especially in bacteria and lower level organisms that have a little bit more leeway in their environmental niche. So they have the same genes, but they produce different genotypes. And so, for instance, this insect, which has high plasticity, switches between developing wings and not developing wings, depending on what kind of habitat it's in, whether or not the leaves on the tree are alive. But actually, it can also switch between being asexual or sexual for its reproduction, depending on whether or not it has enough viable mates. And so you can have organisms like this, which are switching. And so, for instance, if we were to find a fossil of these two organisms encased in something, we wouldn't be sure whether or not they were the same species and a different morph or a different one. Hopefully we would be able to extract DNA. We're getting pretty good at extracting ancient DNA out of things. I was actually at a seminar, and it's crazy the amount of DNA repair we can do and trying to figure it out. Is this it? All right. So one thing I like to do is I like to break up the lecture portion with some videos, and then I like to talk over the videos. So this one I'm not really going to talk over because it's someone explaining something, but this is actually an explanation of a NASA experiment. So what they did was they actually took two people who were twins and they put one in space for a long time. And then they actually looked at the genes that were expressed in these two twins. Nope. Okay, can you move down? I don't think it's gonna.
Two NASA astronauts who also happen to be identical twins.
All right, channel speakers. I just want the screen to be smaller so I can show you more things at once. I don't think this is sharing. Sorry, I'm gonna make this dot disappear because it refuses to, or there we go. All right, when I play this video, can the people online hear it?
Twins. On the left here, this is Mark Kelly.
Yes.
You can see his name on his patch right over there. And then this is Scott Kelly. And the reason why we want to look at these two astronauts in particular is to think about genotype and phenotype that we have been introduced to in other videos. So just as a bit of a review, when people talk about genotype, they're talking about the actual genetic information encoded in an organism's genes. So if you go inside a cell, we have chromosomes, and then stretch of DNA will code for various things, often usually various proteins. But there's a difference between what information is in one's genes versus what actually gets expressed. The observable characteristics, that is phenotype. And two people with the same genotype, they might have a very similar phenotype, but they won't necessarily have an identical phenotype. Because above and beyond the genes affecting phenotype, you also have the environment affecting someone's phenotype. So as you can see here, Mark and Scott Kelly, they're identical twins. They have the same genotype. But if you just look at their facial characteristics, you can see that you could tell the difference between the two. And it's not just the fact that Mark has a mustache and that Scott doesn't. We could draw in a mustache. You would still see that they look different. And that's because their bodies developed in different ways based on the environment that they happen to be in. And this idea that the same genotype could result in variations of phenotype, this is known as phenotype plasticity. Phenotype plasticity. And maybe in one of the most extreme experiments ever conducted on phenotype plasticity, NASA was intrigued to see, well, what would happen to the gene expression if Scott Kelly spent an extensive period of time in space while his twin brother didn't. So they looked at how their bodies expressed certain characteristics before Scott spent an extended period of time in space. And then after he spent that time in space, they studied his body. And they saw that there was differences in the mitochondria, differences in which genes were expressed. In fact, there was a 7% difference in the gene expression after the extended time in space for Scott. So that's an example of that space environment changing Scott's phenotype. And the NASA scientists theorized that it was a low oxygen environment. There was a lot of carbon dioxide in that environment. There were stressors from space travel that would have affected how the genotype, which is identical to his brother Mark's genotype, how it might have been expressed, resulting in the phenotype plasticity above and beyond what other changes in phenotype between Scott and Mark accumulated over the course of their lives, despite having the same genetic information.
Right? So those two scientists, they are, twins, perfect example of exact same DNA producing two different bodies. But those astronauts, one of them was in space, right? So they changed their environment. And so, as they just said, in space, there's not going to be as much gravity. There's not going to be as much oxygen. We're filling spacecrafts with, pure combinations of, oxygen and carbon dioxide, completely different than the mixture of air that you would find on Earth. And so what's going to happen, right? Those cells within its body are going to try to maintain homeostasis. They're going to try to keep themselves in a perfect earthbound, Earth-like atmosphere, but the conditions are different, right? So they're going to have to upregulate. Probably, I would assume that it's similar to when you go on top of a mountain, right? If you know about pro athletes and blood doping, they'll train high up on mountains where there's less oxygen, so their body will naturally start producing more iron and more blood in order to get that extra, those lower levels of oxygen, more of it into their bodies. And so that's probably what happened with that NASA scientist, right? He was in a lower oxygen environment, so his body had to upregulate, which changes that phenotypical expression, those enzymes that he's creating and everything like that. And so that is, a more smaller genetic version of it. But again, this will happen to organisms on a much greater scale, depending on their environment, or if it's happening when they're developing. Bacteria do this a lot too. Bacteria will form colonies and will form biofilms. And so when they form a biofilm, they'll actually change their genetics based on what jobs they need to be accomplished. One of them needs to undergo respiration, and so it will change its outside shell. It'll change the way that it processes oxygen and things like that. But obviously, this biological definition is not the only definition. Once again, there are a lot of subdisciplines in science. So if you're interested in science, I would suggest you take a lot of different courses, and you also, if you can, try to do research in a number of different fields, because scientists look at the world differently based on their scope and where they're coming from. And so one thing, we have this biological one that everyone uses, but there's also this idea of a morphological concept. And this would be probably the earliest and most widely used version of what a species is, right? If you look at early, fossils, look at how we started, before we knew about genes and before we could analyze the genetic code or DNA of something, this is how we decided what a species was. Does it look different than the other one? And obviously these concepts can get very, very specific, but they're distinguished by their physical aspects, right? And again, there's already issues with this between, males and females, egg producers, sperm producers in different species, right? They're going to have physical aspects that are different. And so the nice thing about morphological differences is that, that's applicable to sexual and asexual organisms, right? We can look at what this bacteria looks like. We can look at the surface of its, cell wall or its cell membrane, and we can compare those to other ones, or the way that, it encases its body. But the problem with this one is that importance of features is subjective once again. And so you'll have scientists debate over whether or not certain things like this matter, right? Does the shape of the cell wall matter if all the other genetic information is? Again, this morphological concept kind of mixes up and kind of further messes with that idea of plasticity, right? You know, you can have two bacteria who are working together, cooperating in order to live in a community, a biofilm, and they may have physical features which are different. And so, you know, one thing I'd like to bring up is that none of these definitions are perfect. All of these definitions have some type of pitfall. And so nowadays, I think this is part of why, you know, most science is becoming integrated, right? We have to talk with a lot of different people. We have to come from different avenues and make sure that we're getting all the information we can. And so researchers may disagree on structural differences that are important, especially when we're talking about fossils or things or creatures that are unobservable. And so, you know, this is why there are debates in science. This is why science is not clear cut, especially when we're talking about phylogenetics. And so this definition is good, especially for a lot of other things. It includes bacteria, but we're starting to combine these. And so a lot of papers nowadays are going to be using both genetic and morphological differences in order to discern what is a new species. Now, there's also an ecological concept. I did say that the biological one was more of an ecological concept, but when we mention this ecological concept, we're more talking about food webs. We're talking about places and roles in an environment. You know, so for instance, there's this badger type organism that's eating Timmy Turner's dad's sandwich. You know, there are organisms which will actually change their, you know, niche in an environment, right? There are, for instance, we discovered a new species of mosquito that was actually, instead of laying its eggs inside, you know, pools of water or lakes or some of these other environments, they actually learned how to utilize man-made structures. So they started figuring out, oh, I'm going to use this waste barrel or these different human-made, non-biologically relevant areas, and they're going to start only breeding in those areas. Why? Because people leave stuff around. People aren't messing with, say, you know, on a construction site, these mosquitoes will just lay eggs on the top of this barrel that's caught water. And that actually led to a genetic change in that species. And that species is now, say, only mating in that certain area or in those those man-made structures. And so they actually become different in that way. And so so looking at the ecological concept, we're worried about the ecological niche, right? The sum of how these species, not spices, interact with both living and non-living parts. Yes, there will be typos, don't worry. So these spices interact with both living and non-living things. They provide different nutrients to organisms above them, and they also take in different ones. And so, you know, they may look similar, but partake in completely different lifestyles. So, you know, think about, you know, I'm saying spiders because that's what I like to study. But, you know, spiders will be very similar sizes. You know, they could even have the same niche in terms of they are web makers, right? There are spiders that'll make webs on your porch, but there are also spiders that'll make webs in between trees. Sometimes these differences are just lifestyle. Say a spider is nocturnal versus diurnal. It may be the same size, it may look very similar, but it comes out at a different time in the day. Or, you know, often spiders are gradiated based on their size and where they make webs. So for instance, there are spiders that are smaller that'll make webs, say on the bottom of leaf matter, and then bigger ones who aren't afraid of being eaten will make theirs at the top of plants, right? If you go out to the preserve, if you go out and walk around in the grass there, you'll find tons of spiders in the exact same environment, seemingly making the same web structure, but are completely different species because they're going to be eating different things at different time in the day. They're going to be eaten by different organisms. They're doing a completely different job in the environment. And so, you know, problem with this, plants are a big hassle because plants can cross-pollinate, they can self-pollinate, you know, they may confuse and create lots of different types of hybrids. When I talk about hybrids in terms of in terms of animals, it'll be a little bit more apparent about how this can be like a dead end evolution. But plants can also cause a lot of confusion, especially because plants have a lot more chromosomes than we do. So, their DNA changes in a much more robust way, depending on the species. But ecological niche, this is probably most useful for those asexual species, right? Once again, what are those bacteria providing in that environment? Is it changing? Do we have a different species of staph? Does the staph bacteria choose to live on certain things or can only grow off other things? And so we have these different methods of speciation, right? So we have different ways that organisms are being isolated from one another. And so these different methods of speciation can kind of explain, these lifestyle differences. So for instance, the idea of allopatric speciation, which is the separation of species based on geographic isolation. What's the easiest way to have reproductive isolation? Completely block those organisms from living near one another. And so my example of this, though, is this spider here on the right, which is called Lorenzioides. It's one of the most common spiders. It's found pretty much everywhere. If you have webs on your porch or something, it's probably this species. If you look at the map at the top of your screen, all of those green spots are areas in which Lorenzioides exist. So this species is not only throughout most of America, it's in Canada, it's throughout all of Europe, it's even all the way in Australia, if you look at that little area, and on the eastern coast of China and Russia. So these species are geographically isolated, but they are the exact same species. And so what's happening? Well, there needs to be a selection pressure, right, in order to force a change in genes or in behavior. Remember, we were talking about things such as founder's effect, talking about these different ways that organisms populations can be isolated from one another. But if there's no change in the selection pressure, if that adaptation that you have is still evolutionarily viable in your new environment, you're not going to change, right? So there needs to be some shift. And so if you look at, besides the Australian population, most of these spiders are in the same, latitude, longitude range, right? They don't really come down beyond that one area. And obviously there are differences based on, geography and height and stuff like that. But it's very important for these organisms to have some type of change in their environment in order to cause this type of speciation. And so, these organisms might be widespread because they existed when Pangea was a thing. So all these areas were together and then they separated. Or you could be having one cool thing that spiders do is they actually can fly using their silk, especially if they're really small. So if you ever seen Charlotte's Web at the end, Charlotte dies and all her babies are like floating away. That's like a real thing that spiders can do, especially babies. They'll release silk and they're so light that the silk actually gets grabbed by the air currents and it picks them up and they can fly. And so it is possible, they found them miles in the air, that like spiders can actually actively float in the air from America to Europe, these depending on the currents. And so, these different species are only going to develop when there's a different niche. There's a different type of pressure that's put upon them. You can also have the speciation that's occurring based on genetics instead of separation. So divergent populations, obviously evolution still occurs even if you don't separate organisms, right? Organisms in an environment who are closely coming into contact with each other, if they live in the same geographical area, can still turn into new species, right? Not everything is because it is isolated. But how does that occur? And so one thing, a good example of how these speciation occurs is in, say, plants and strawberries we'll talk about. So for instance, strawberries are what are known as polyploid, which means that they have way more types of chromosomes than ours. We're diploid. They have, depending on the species, they can have anywhere between 4 pairs of chromosomes to up to 8. And so they're going to have a much more complex genetic system. But what's going to happen is some traits are actually decided by the number of copies of a said chromosome that you have. So for instance, when we have normal fruit, so for instance, wild strawberries in that picture on the bottom are much, much smaller than cultivated strawberries that we have. And why is that? Is because we have a lot more chromosomes that are stuffed into that strawberry. And so if you remember talking about like genetic information being passed down, how there can be errors within that DNA replication, errors in that DNA being passed on. Sometimes if there's an accident in cell division, maybe those chromosomes don't break apart. You can have a different number of chromosomes that actually get passed off to your offspring. Sometimes that is lethal and the organism will just die. Sometimes it won't for some reason or another. So for instance, when we breed these strawberries to be polyploid, to have even more sets of genomes, we actually have them expressing hyper, oh God, I'm blanking on the hormone that causes fruit growth. I'm blanking on it right now, I'm sorry, but it causes hyper expression of that. And so we have much larger strawberries. But in a much more applicable to humans type of evolutionary way, if you track the changes in chromosomes over time, chimps actually have 1 chromosome more than we do. And so do a lot of the great apes, or so do the great apes. And so what we believe happened was during the evolution of chimps, there was a point where 2 of their chromosomes did not break apart and they actually became 1. And so we have on our, I think it's 22nd chromosome, we actually have all of the genes that are in the chimp, phenotype. If you remember, we share like 99.6 or something % of our genome with chimps. The thing is though, our chromosomes are different. Our chromosome placement is different. And so that could actually cause us to no longer be able to mate with chimps. Obviously, our genomes aren't the same. If we were to say mix our DNA together, I'm not sure if anyone's tried what happens when you mix human and chimp DNA. I'm pretty sure what'll happen is the lack of consistency in chromosomes is going to cause that new cell, that zygote that's formed, to die and be unable to properly undergo its processes. And so, these accidents can often lead to death of an organism, but sometimes, depending on exactly which chromosomes are mixed and how important those chromosomes are to different biological functions, you're going to have the possibility of a new species erupting from that. But as I said, it's more commonplace in plants, where you have plants that are cross-pollinating, you have plants that are sharing genomes a lot more often. And so that leads again to the overexpression of certain genes, which again will make a new species depending on what it is. Cool. So, and then there's this idea of alliploid. So, there are hybrids. So I mentioned that before, right? There are a lot of hybrids. There are organisms that we've been mating together just to see what they do. For instance, we have ligers, which are bred for their skills in magic. But, the problem is that often these are dead end genomes because the once, perfectly fertile organism is now sterile once it's been mated with something else. So for instance, I always forget what the difference is. You have Tigons and liger. The difference is which one is the mother. I'm pretty sure a Tigon is when you have a tiger mother and then a sperm donor of a lion, and then the liger would be the opposite. But one that you've heard of and seen all the time is a mule. A mule is a horse and a donkey mixed together. They seem like very closely related organisms, and again, you can mate them together and you will get offspring. The problem is that tigon, ligers, and mules are all sterile. You know, they are when the zygote is formed and it starts replicating, it is able to get up to this form. But because of the way that the chromosomes work, those offspring, those sperm and those egg cells that are produced cannot mix with another. And so there are different ways that these hybrids can kind of be dead ends. You know, sometimes they don't produce sperm at all. Sometimes their sperm is just completely, you know, incompatible with the egg of the higher species. If you were to say mate them together, you're still not going to get something that's going to be viable. Cool. And so these are organisms that are called as allopolyploid. So they are individuals that have two sets of chromosomes from more than one species. And obviously, you know, this doesn't always work out in terms of stuff like that. Cool. So my next video is going to be speciation through habitat change. So we're going to talk about these marine iguanas in the Galapagos. So really cool thing. Iguanas, usually not big swimmers, usually are living on land, living on trees. You know, if you are living on trees, they're living in warm environments, right? You know, when it started getting below 50 degrees in Florida this past year, they started falling out of trees because they were too cold, that they were hibernating. But in the Galapagos, there's these species of marine iguanas who actually, they're not sure how they got there because the Galapagos Islands are really, really isolated. You know, one of the reasons that Darwin picked them in order to study was because they were so isolated and very few organisms are capable of migrating there. But at some point, one of these iguanas from the mainland got isolated and pushed off into the water and wound up in the Galapagos. And so you can see how this video is going to talk about physical changes that occurred and then just very, very different environmental niche that this organism holds compared to other iguanas. What on earth is that? What did I do?
Of these iguanas almost certainly lived in the jungles of Central America. They're still in the trees overhanging the rivers, nibbling leaves, or on rafts of reeds. Just occasionally are swept out to sea, and the vast majority, of course, die there. But just a few, a long time ago, were fortunate enough to be swept by favorable currents out to the ocean and pitched up here.
In their ancestral rainforest habitat, iguanas are vegetarians. Here, they browse on juicy leaves. But the iguanas that first appeared in the Galapagos could find no such things. So these iguanas, to survive, had to eat the only kind of leaf that was available: seaweed. And to get the best of that, they had to do something even more radical. They had to swim. They even learned to dive. They acquired the ability to hold their breath for up to an hour, so that they could swim down to a depth of 20 meters. Their claws strengthened, so they could cling to the rocks on the seabed. And under the water, they found an endless supply of seaweed, which grew in abundance in the nutrient-rich currents that flow around the islands. But that was not all. Their snouts became flatter to help them graze. And their teeth became sharper to grip the slippery seaweed.
Yeah. David Attenborough. I love him. He's so good. All right. Right. So you had these organisms that were separated, and because they were separated, they wound up in a new environment, and they had to form an entirely new environmental niche, right? They had to eat new things. They had to learn to survive in an aquatic environment versus another one. And so these organisms look mostly the same. You know, if you're just staring at a lizard, you might not notice the difference. But evolutionarily, this separation caused them to go to a new environment and then have to adapt to new physical traits that had to be applicable in order to survive in this environment, right? As I said, shorter snouts so they could eat, thicker claws, different types of teeth. Even if you look at them, their coloration is completely different, right? They no longer are living in a lush green environment. They're swimming around. So their colors actually don't matter as much, at least in terms of being green. One thing that is cool though, is that if you look at a lot of deep sea life are browns and reds. And so the reason that is because red light, being low energy and really long lengths, is actually really bad at penetrating into depths of water. And so organisms that are, you know, blue and purple are a lot easier and a lot brighter underwater. And so these organisms went from being green, which would be much brighter, much higher energy in the water, to being red and brown and black. And so those colors are going to blend in more with just the darkness that's in the water. I sadly don't have a picture of it, but you can look it up. There are certain scientists who do experiments on coloration. There's a gentleman known as Sanka Johnson, who also does bioluminescence down at Duke. And he specifically takes pictures of things underwater at visible light, red light, and blue light. And you can see how, you know, a coral reef, which is unbelievably beautiful, when you look at it under red light, it's much duller. It's much harder to see everything there. Things are hiding better. And so organisms are obviously going to evolve in a way that makes it more, makes their adaptation much better in an environment. And so as an example, this is from your textbook. I think it's from an older version of your textbook though. So they probably updated, it'll probably look a little bit different. But it's showing two examples of how this evolution occurs, right? So on the left, you have separation of a species from a creation of a species through separation, geographic isolation, right? So you have this river, this lake, and then, you know, it starts to dry up. As the water dries up, those fish are no longer going to be accessible to both parts of that lake. And so they're going to separate. And so over a long time, these organisms might develop new genetic material, right? They might change for their environment. You know, it might just randomly, genetic drift, for instance, could be something, right? Maybe the organisms that get isolated on the left are a slightly different color. And so now the population on the left are blue, whereas the other ones are orange. Then you have, the one on the right, which is not really super explanatory, but the sympatric evolution, sympatric. So there's, the subset of the population forms a new species without geographic isolation. And so one cool thing that happens is sometimes in the environment, we have this occur where on the left, you have, say, a river dry up or something, and all of a sudden these two bodies of water are separated. And then we have what's happening now where sea levels rise. And those two populations that were separated are now coming back into contact with one another. And so they could either exist as different species, or depending on how genetically isolated they've been over these years, they might start to mate again and actually create hybrids. And so nature, we're not the only ones who are mixing tigers and ligons and making these different species. A lot of fish don't know any better, right? For instance, there's an example in the Great Lakes where two species of fish got separated. They came from a common ancestor. They developed into new species. The Great Lakes start to fill up a little bit more, and then they came into contact with one another. And when that happened, primarily the difference was coloration, right? There's this blue and this orange fish. They might not, mate with each other because they notice you're not the right color. But in the Great Lakes, what happened was not only did the water level rise, but it started to flow a little bit harder. And so you had this environment that became very murky and dirty. And so the fish could no longer see as well. And so this coloration difference didn't matter at all. And so those species started to mate once again, and then they actually became the same species once again. So they diverged into two new species, came back into contact, and then became one species again. And so there can be very, very complicated relationships between organisms and these type of hybrids. It's not as simple as hybrids are useless or hybrids create new species. It really, like all of these things, all of it is a case-by-case basis. And so how long does it take for species to evolve? It really depends. For instance, we believe that most species take 5.5 million years, but sometimes it can only take 500,000 years. That example that I gave you about the fish in the Great Lakes only took 4,000 years. So, if organisms are very closely related, sometimes it doesn't happen. But for instance, another example is the most diverse species and populous species on the planet are beetles. Beetles, most insects are beetles, and then most sea creatures become crabs. There's something about having a short, sturdy body that is very well protected. And so, for instance, beetles have been doing great. They really don't change very often. It can take 40 million years for beetles to develop into another one. Beetless. And so one of the issues is, again, looking at fossils, is that we have limited information, right? And so there's this idea about punctuated equilibria. And so if you look at the fossil record, one thing that we've noticed is that you will have long periods in which organisms will basically never change physically, or very, very small changes. And then all of a sudden, there's a huge change in these organisms. And again, this is because of fossils, right? We can't see these intermediate organisms. And so what's happening is we're only going to see the most extreme changes, or those extreme changes that have actually been encased. And so when we're looking at fossils, this is when we can tell speciation occurs, right? We can tell speciation is occurring because of these drastic physical difference in the fossil records. But again, it's hard to gauge because of the lack of information, this inability to look at missing links, or the inability to tell subtle differences in the fossil record. Again, there are more than just differences in bone structure. There could be differences in coloration, right? Think of how similar body plans birds can have, but their completely different coloration is what is going to stop them from mating with one another. And again, they could be very, very subtle in these differences. We're getting better at that. But again, it's a limiting factor. And so it also has to do with our ability to look at time frames in sediment settling, right? Different areas of the world have different levels of rain, different levels of erosion. And so sediment levels can be much more compact or much more spread out depending on where you are. And obviously, again, our information is getting better every year. We're able to look at genetic information. We're starting to be able to look at, again, DNA from fossils. And so hopefully we can fill in these gaps. But, you know, these patterns of evolution are very, very difficult, right? So we see mostly this thing at the top, right? Punctuated patterns. We see species A, and then we see all of a sudden it becomes species B and C. And then we see that species for the rest of time or the rest of the fossil record. But the thing is that it really is, for the most part, this part on the bottom, right? For the most part, organisms are undergoing this gradual change. Genetic changes, those alleles in the environment are changing over generations, and they're going to be changing in slow amounts, right? Natural selection is slowly gonna, take out this brown and tan moth, and it's gonna start making it more advantageous to be full white or full, say, orange, depending again on your environment and the way you're moving. Let's see, great. So hybrids, what happens when we bring when hybridization occurs in the environment, right? And so hybrids are, trying to create, hybrids are kind of like this punctuated evolution, right? You're just combining them and then you have this new species. We hope that it's fertile, it never is. And then we move on, right? But hybrids can happen, can be limited for a number of reasons, right? If there was no limitations, right, organisms would just hybridize all the time and you would just constantly have new species, right? All of this genetic information is being exchanged. Why are we not coming up with new species all the time, every day? Why are, birds not mixing and matching? And that's because there are a lot of limitations to hybridization. And so the last thing I'll touch on a little bit today is these hybrids and how hybridization, what limits hybridization, right? So there are two different sets that we're going to talk about. There are prezygotic and postzygotic barriers. And so prezygotic are all the barriers that can limit two species mating with one another before they actually ever reproduce. Postzygotic is those different barriers that happen once fertilization occurs. So once the sperm and egg come together, why don't they lead to a new species? And so prezygotic barriers have to do with a lot of stopping members from mating. Why are they not physically mating? Well, there could be a lot of different ways. There could be sexual selection, right? So you could have mating which is done based on some type of female choice. And I say that because in the environment, female egg producers are generally the ones who choose their mates in most animal kingdoms. They're the ones who have to carry the babies. They're the ones who are producing all the eggs. They're making a selection of what sperm to, what is the most advantageous for it. But yeah, they're stopping members from mating. So say you're not doing the right mating dance, or you don't look like you're the right color for our species, though that may just innately stop that. There's also the idea of preventing mating from being successful. You could just not have the right tools. So something super simple is I always forget the Latin name for them, but there are two species of snails. And essentially the only difference between these species is that their shells curve in slightly different ways. One curves to the left and one curves to the right. And so what happens is, because of the way their shells are lined up, if one curves to the left and one curves to the right, they actually can't mate because their genitals can't get close enough in order to actually impregnate the other one. So if you don't have the right tools, spiders have special lock and key mechanisms. The male spider have these pedipalps, which are essentially little boxing gloves with like crazy keys at the end. And so if a male spider tries to mate with the wrong female, that key is not going to open up her uterus, and he's not going to be able to inject his sperm into it. And so, you know, once you evolve and your tool changes, or your method of changing, of selecting mates changes, you may not be able to no longer, you know, do that. So that's just going to stop species from mating of different ones. You then have hindered fertilization. Again, it could be a difference in the way you're using tools, the way that your genitals match, or, you know, for instance, again, spiders, male spiders, sperm are actually immotile. They are encased in a special coating that keeps them from degrading, and they actually don't break down until the female actually starts creating the appropriate enzymes to actually break down the casing on that sperm and then allow them to come out. And so, for instance, you know, if a male is to impregnate the wrong female and she doesn't have the enzymes in order to open up that sperm casing, she's not going to be able to impregnate her egg, be able to fertilize her eggs. And a cool thing about spiders is that females can actually mate with multiple males and hold on to the sperm inside of them, and then they decide when they feel like fertilizing their eggs. And so again, if they mated with the wrong species and they don't have the enzymes to break down that casing, fertilization never occurs. But, you know, as I'm saying, there are post-zygonic barriers. So even if you are capable of mating with one another, doesn't necessarily mean it's going to work out. And so there's just genetic compatibility issues. Generally speaking, a lot of fertilized zygotes will just die. You know, think about it. If you have the wrong number of chromosomes, when your chromosomes go to pair up, you're going to have excess amounts. You're going to have different issues of that. Perhaps, you know, as that zygote is starting to grow up, you're missing some important enzyme, right? You're missing an important enzyme that helps break down hydrogen peroxide. And so your cell, as it's starting to develop, produces hydrogen peroxide. Because of the way that your chromosomes came together, you don't have the enzyme to break that down. Your cell's going to die from overexposure to hydrogen peroxide. You know, and so there are so many different, you know, that's just one example. There's so many different ways that you can have genetic incompatibility in that way, or can be missing certain genes. Perhaps, you know, when they come together, you're actually missing one of the genes that helps regulate control, right? If you have different body plans, you could have different sets of chromosomes which are trying to accomplish different, you know, different things, right? You know, creation of the jaw structure of ours is so different from that of a chimp, right? If cells are starting to specialize, they're starting to try to turn into different parts of the animal. If your plans aren't lined up, it may not work. And then one of the big things that we were talking about is this idea of just offspring sterility, right? A lot of offspring, when they are mixed, are not capable of making zygotes, right? When you go through mitosis, when you go through meiosis, right? As meiosis is occurring, all your chromosomes are supposed to line up, and then they, you know, when it's applicable, they shift to the edges, you start having those cells breaking apart, cytokinesis and all of that jazz. But if you don't have the right chromosomes to line up, or the chromosomes aren't splitting properly, that organism is not going to be able to produce those zygotes. Meiosis is not going to be able to properly go out. Or, you know, again, if you're missing chromosomes, maybe you undergo meiosis, but the, you know, sperm that you're passing on don't have enough chromosomes. And so when they come together with another egg, or, you know, or with sperm, they're not actually going to be able to go forward. And then there's one that I find really interesting, which is the idea of hybrid breakdown. So sometimes when hybrids are created, that first generation of hybrids is actually fertile, and they're able to mate with themselves and or the parents. But there's something about this second, generation, that F2 generation, if you're thinking of Mendelian genetics, in which it stops working, in which some aspect of the genetic code, after they get broken down again and split, stop working. And so all of a sudden you had what you think is a viable hybrid. That second generation though isn't fertile or starts to die out and becomes weaker. And so this is a nice graphic from your textbook. I like this one because it shows all of the things I was just talking about in a nice timeline, right? So on the left, you have different things such as different things that are the, the pre-zygotic. So do these organisms have different habitat, right? Once again, are these snakes, living on land versus water, not going to come into contact with each other? Temporal isolation, time of the day. Again, are you diurnal versus nocturnal? You could be very similar species, such as these two skunks, badgers, skunks. These two skunks, and simply because you don't come into contact with one another, you're not going to mate. Then you have behavioral differences, such as courtship dances. What is this one? Oh, yeah. Yeah, so, no. Yeah, you could have differences in genetic information or the tools. So for instance, these are those two snails I was talking about. If you look, because of the way their shells are oriented, if you look at, say, this one here and this one here, they're not going to be able to mate because one is bending that way and one is bending that way. And then you have these in green, you have those post-zygotic barriers, right? So you have just viability, genes of different parents may interact and just aren't creating something that's survivability. So one of those things could be, for instance, this salamander here, and next class I'll show you another video because it's 10:15, so I'm going to wrap it up for today. And that is, for instance, you have species A, which is black and very good at blending in with the environment. You have species B, which is really bright and red, and it looks toxic. So nothing wants to eat it because they're afraid that it's going to kill them, right? When you create a hybrid of those two, for instance, this species, it's both black and red. It's no longer looks deadly enough that something's going to ignore it. So it's going to get eaten. And now that it's bright red, it no longer blends in with the environment around it. And so sometimes just having a mixture of these two types of properties, or both having a phenotypical mixture of two organisms, makes it so that you are no longer advantageous. And so that could just be a reason. Maybe those salamanders are viable. They can produce fertile offspring. They're non-sterile, and maybe they'll even mate with each other or other ones. And so that could continue, but they're being picked off too readily and they die. So their genome is never fully expressed in the population. So it's not always a limiting factor on physical genetic information. Sometimes the environment is just not going to let you evolve. Natural selection is going to take you out way too quickly. Cool. All right. So for this, it's been recorded. I'm going to put this up on Moodle. I'm also going to put a link on Moodle to my YouTube page. And so those talks will also be up on YouTube. I will have also, though, talks from that from 107. So there'll be a few, but this should be the most. All right. Thank you all for being here. Thank you all for coming.
Thank you.
Thank you.
Thank you.